Okay, we're going to go ahead and get started. Again, my name is Ashley Givens. I am the operations coordinator for the Ignite team. Welcome to the Ignite Q&A webinar. Today, I'm going to introduce our panelists that we have today. We have Dr. Rebecca Roof, who is the program director. We have Dr. Julia Bachman, who is our health program specialist, Dr. Shardo Spriggs, who is our program manager, and we have a special guest from NCCIH, Dr. Craig Hopp. Okay, how today's webinar is going to work. This webinar will be recorded. The webinar recording and slides will be posted on the Ignite website, and we will also send you the slides after the webinar is finished. Also, please use, there's a Q&A box at the bottom of your screen. Please use that box to post all questions, and we will respond to those Q&A questions after the presentation is finished. If you have project-specific questions, it would be better if you would, it actually will help us if you would send us your AIMS page, and they can set up a call so you guys can talk further about your project-specific questions. All right, so thank you again, and we're going to go ahead and get started, and I'm going to turn the presentation over to Dr. Roof at this time.
Great, thank you, Ashley. This is Becky Roof. I manage the Ignite program. I'm excited to tell you today about innovation grants to nurture initial translational efforts, our early stage translational program. As Ashley said, you'll be getting these slides, and there's some links in there, so you'll be getting those later. And we'll talk for a little bit, and then we'll have time for Q&A. So first of all, let me advance my slides. Okay, there we go. This is how you know we're live, folks. All right, so first of all, the goal of Ignite is an early stage therapy development program. The goal is to get you ready for the next stage, right? These FOAs were written such that they would help you get the entry criteria for later stage Blueprint Neurotherapeutics program for small molecules. And now we have this new BPN for Biologics program. They have a very similar entry criteria. It would be a really good idea as you're thinking about an Ignite grant to look at the entry criteria for these programs and make sure that when you write your Ignite grant that you propose what you need to do to get yourself to the next program, right? You want to make sure that you use those 3 years to meet those entry criteria so that you're ready to move forward at the end. Okay, this program is very much designed to be a feeder program for this later stage. All right, so we have 3 different FOAs in Ignite listed here. We're going to talk about each of these in detail, but let me just give you the overview. The first is PIR 21-124 for assay development and therapeutic agent identification. The second one is PIR 21-123 for development and validation of model systems. And the third one is PIR 21-122 for neurotherapeutic agent characterization and in vivo efficacy studies. All 3 of these FOAs have the same budget and the same time period. They're 3-year grants. They are a budget limit of $750 direct costs for the full 3 years with no more than $499 in any given year. Our next due date is June 17th, and after that is October. Every round I seem to get a question. Somebody says, I'm eligible for continuous submission. Does that apply here? The answer is no, but under this notice listed here, you might be eligible to apply late, but take a good look at it and make sure and realize that we can't pre-approve anything. So if you can get your application in on time, that's all the better. I will also mention there is not a special A1 date. So if you're doing a resubmission, it's the same due date as if you're doing a new application. So please be aware of that. All right. Now I'm going to talk for just a minute about the R61r33 mechanism. I realize this is new for some people. I want you to realize this is not an R01 type mechanism. This is a little bit different. So I'm going to take a minute to walk you through what that looks like so that you are prepared. So the first phase is the R61, where you demonstrate feasibility, you get prepared. This is where you get your ducks in a row. This is where you validate the assay that you're going to use later for your high throughput screen, or maybe you optimize compounds that you're going to use later for your proof of concept efficacy study. This is, setting the stage, showing that it's feasible, showing that it's worth going forward. Okay. Then you will have a set of go, no go milestones. These milestones need to be clear and quantitative. If these milestones are met, you will move forward to the R33 stage. We call this the main event, but it could be, it could be your high throughput screen, it could be your proof of concept study, it could be your external validation of your animal model. Whatever it is you need, that killer that you need to get you the entry criteria for the next stage program. Okay. It is extremely important. It is essential that the milestones be clear and quantitative. All right. There is a single go, no go decision point between the two phases. There are other mechanisms at NIH with multiple milestones. This has only one. And then the transition to the R33 is via an administrative review. Now, what that means is that the decision is made internal at NIH. We do not send them back out to peer review. And the benefit of that is that it can happen much more quickly. If everything's straightforward and your paperwork is done on time, we can go from the R61 to the R33 without any lapse of funding. Okay, and the reason that we can do this via an administrative review is because review panel will see your milestones before you get funded when you apply. And if they are really clear and they're really quantitative and really definitive, there should be no confusion about whether or not your milestones are met. It should be a pretty obvious decision, which is why we can do it internally via administrative review. But that's why your milestones need to be really clear. Now, why would anyone even do a milestone grant? The benefit here is that it allows you to propose riskier science. You can propose something where phase 2 completely depends on phase 1 working. That kind of stuff is sometimes hard to get funded in an R01. But here, review is perfectly happy to give that kind of work a good score. We're perfectly happy to fund it. It's actually expected. It's by design. Phase 2 depends on phase 1 working. And this milestone is what makes this whole mechanism work. Okay. So there is the benefit there that you can propose riskier science. And we can fund riskier science. So that's why we use these mechanisms. All right, so I'm going to harp on this milestone thing for just a minute more. And the reason is that a lot of people struggle with this. This is something that gets dinged in review quite a lot. So I just want to really make sure that I help you guys prevent having that problem. Especially if you've not written milestones before, it can be, it can be a little bit challenging. Milestones are not a list of activities that you need to do. They're not, things that are such a low bar that you've already met them. They really do need to reflect success of the first phase and that it's worth going forward. All right, so I'm going to give you some examples here. This is a weak milestone. We're going to scale up a compound. A stronger milestone is we need X grams, we need this purity. This is how we're going to define it. Okay, you can see the difference. A weak milestone might be we're going to do PK studies on a bunch of compounds and pick the best one. A stronger milestone is this is what we need to see. We need to see these properties. We need to see this penetration or this half-life or whatever is appropriate for your study. Okay, you can see how that's more definitive, and you can see where when it comes time to evaluate your milestones, it's going to be really easy to see whether this milestone is met or not. Okay, a weak milestone is we'll have fully optimized and validated an assay. Stronger version is, we need to see this Z prime. We need to see this signal to noise or whatever. And I don't want to dictate milestones here. I don't want you to think that NIH is requiring me to look at half-life or Z prime or whatever. I want you to propose what is appropriate for your studies. I'm just illustrating here the difference between a weak milestone and a stronger milestone. All right, the last one is, treatment of our protein is going to decrease seizures. That's a weak milestone. A stronger milestone is we need to decrease seizures by this much. We need to see it for this long. And this is our N, by the way, and this is our control. Another thing that people sometimes do that review will pick on is they'll say we need to see a statistically significant difference compared to control. And that is quantitative, but it might not be biologically meaningful. So if you can define it rather than just say statistically significant, that often does better. And when you define it, that you explain where it came from. In this example, you need to see a decrease in seizure frequency by X. How do you know X is meaningful? If a patient had that much fewer seizures, would they consider that beneficial? Right, and justify that. So we have more examples on our website. Hopefully you found these examples to be helpful. Really I'm just trying to illustrate what quantitative looks like, what clear looks like, and so that you know what we're talking about there. Our review panels are really, really good at looking at this. So know that that's coming and be prepared. Okay, so now we're going to walk through each of these FOAs one by one. The first one is PAR 21-124, which is the assay development one. The goals here really are to develop new assays and use those assays for screening of new agents to treat for therapeutic development. The entry criteria, they need to be novel, but also needs to have a good, strong biological rationale. Okay, so there's a balance there. Relevance for therapeutic development and availability of agents or library. I will say a lot of people use this FOA for high throughput screening, but it does not have to be. If you want to do smaller screens and you have rationale for why that's going to work, that's perfectly acceptable as well. All right, so some things you might do in your first phase include the following, but are not limited to. Development of assays to support a screening funnel, or development of assays for future ex vivo, in vitro efficacy, potency studies. You might look at assays to look at selectivity of your compounds, or develop assays for your high throughput screen if that's what you're doing. Obviously, a combination of assays might be useful, and that is certainly supported. You might transition from the R61 to the R33 when you can say that your assays are fully developed and optimized, fully validated. If maybe you need to do some physiochemical or characterization of your test compounds. Maybe you need to make cell lines or vectors to produce your agent, especially if it's some kind of biologic, right? Those are things that you might say that you need to do in order to complete the R61. Some things you might do in the R33 is, obviously screening. You might either a high throughput screen or a not high throughput screen. You might prepare your therapeutic agents. You might confirm the structure. You might do some optimization. Property characterization, PK, ADME kind of work would be appropriate. In vitro PK. You might look at off-target activities. You might do some optimization. For example, okay, including but not limited to. So now I'm going to have Craig talk to you for just a minute about what NCCIH is looking for in these FOAs. Craig?
Yes, thanks, Becky. So I'm Craig Hopp. I'm with NCCIH, and we are participating in this IGNITE program. This actually only in this particular PAR. And our interests obviously are a subset of those of the overall IGNITE program and of NINDS. NCCIH interests are limited for the purposes of this PAR to development of analgesic assays, and that obviously could be lots of different things, and the use of natural products as a screen in those assays. And so all the things that Becky just said about the types of research that could be done in the R61 and R33 phases are still appropriate for NCCIH as long as you're focused on natural products for an analgesic target. I will add here that the size of the library, there's no predefined sort of how big does a library have to be, but it does have to be exclusively natural products. If it's one of these mixed libraries that has a diversity panel of things, some are natural products and some are synthetics, then that would not be appropriate for NCCIH. We really are only interested in natural products. And I will even extend that to say some libraries have natural products as well as sort of, I would say, semi-synthetic derivatives of those natural products. Those would not be appropriate for NCCH either. Now, they might still be appropriate for NINDS, and I'll let Becky sort of field any questions about that. But anyway, that's, I guess, is the broad scopes of what NCCH is interested in for the purposes of this initiative.
Great, thanks, Craig. Nick, moving on, we'll talk about what's non-responsive. Now, I will point out one thing that is new with these new reissues of Ignite is we now have things, we will withdraw things that are not responsive. So it's really important that you take a look at these activities. Contact us if you have questions. It's not our goal to be, throwing projects away. We really want to help you propose something that's going to be appropriate so that we can have a successful application. Okay, so applications that are focused on indications outside of the mission of NINDS or outside NCCIH's interest, as you just heard about, will be considered non-responsive. Okay, in general, things are going to be non-responsive if they're basic science, if they're on the other end, if they're better suited for blueprint. Things are going to be considered non-responsive if they're a better fit for our biomarkers or a devices program. Each of these Ignite FOAs has their zone, and if it's too early or too late, or if it's a fit for another FOA, we're going to consider that non-responsive. So just pay attention, make sure you're applying to the right FOA. I'll read through this list, but because I want you to be aware, but please pay attention to this. Development of risk detection, diagnostic, prognostic, predictive, preventative biomarkers, or applications with the primary focus PD biomarkers will be non-responsive. Device development, non-responsive. Assays or probes to support basic understanding of basic science, target identification, studies of disease mechanisms. These things can be supported just fine in the R01 space, so they're out of scope for Ignite. In vivo efficacy studies, you'll hear about ways to get that funded later on in our talk today. Clinical compound manufacture, development of bioassays for clinical use. Those are, if you're ready for clinical use, you're beyond Ignite. IND enabling studies, that's better suited for blueprint stage projects. And human subjects research, unless it meets the E4 criteria. And Jules will tell us a little bit more about human subjects criteria later on. Okay, so now I'm going to turn it over to my colleague. I believe Chardell is covering this one. Take it away.
No, I'm doing this one.
Oh, I'm sorry, Jules.
Okay. Hi, everyone. I am Julia Bachman. The second funding opportunity in Ignite is PAR 21-123, which is for the development and validation of model systems. I should point out that prior issues of this funding opportunity did include the development of pharmacodynamic biomarkers as well as model systems, but PD markers as a primary intent is no longer a focus of this FOA. So the goal of this funding opportunity is to promote a significant improvement in the translational relevance of animal models and ex vivo systems that will be utilized to facilitate the development of neurotherapeutics. So essentially, we're looking to provide translational tools for the drug discovery and development process. And, not something that's just to be used in your lab, but something that would really make a difference for the whole disease area. Something that other investigators or companies would want to use as they're developing candidate neurotherapeutics as well. So in addition to having a strong biological and translational rationale for your model, there should also be evidence of value for future drug discovery and development. Next slide, please. Becky, can you advance the slide? Thanks. So two important concepts to define for the context of this funding opportunity are internal and external validation. Internal validation addresses the consistency of the experimental design. It involves characterizing the precision, reliability, analytical sensitivity, accuracy, and dynamic range of the endpoints used to assess the animal model or model system. whereas external validation refers to the extent to which results of a study can be generalized from one setting to another, namely the preclinical to clinical space. And these are typically thought of as three different types of validities. Face validity refers to the similarity between the model and clinical manifestation of the disease, such as symptoms and morphology and so forth. Construct validity is the similarity between the model or model system and the physiological basis of the human disorder, so in terms of what is known about the disease mechanism. And finally, predictive validity is the similarity between the effect of a validated therapeutic intervention in the model and in the clinical disease population. Next slide. Examples of activities for the R61 phase of this funding opportunity include the initial development of the model or ex vivo system. And you can completely develop and validate a model from scratch, or you can take a model that has not been well validated and validate it. Other activities include any optimization related to the feasibility, endpoint range or sensitivity, et cetera, and internal validation for the endpoints used, as well as any scale up needed for the R33 phase. For example, would be if you're developing a genetically modified animal, you'd need to make sure that the breeding is sufficient and you have a stable generation to work from. So by the end of the R61 phase, you want to be able to show that your model or model system is feasible for the external validation activities in the R33, whether that be the actual ends for your animals or the basic internal validation studies for your endpoints. You also want to show that it reflects the functional or physiological pathway of the disease as intended. And again, your milestones should address these aspects in a quantitative way. So examples of activities for the R33 phase are all of the external validation studies, including comparisons of the model phenotype to human disease, comparisons of disease etiology in preclinical species to what is known about the human disease, or comparing the efficacy of a clinically validated therapeutic agent in the new model system if such therapeutics are available. And I do want to stress that because there are plenty of neurological disorders that don't yet have validated therapeutics. So it's not necessarily expected that you're going to be able to provide predictive validity for your model in this case. But overall, I do again want to emphasize that we want a very rigorous approach that is going to be translational. So i.e. the model will mimic as much as possible the human disease or condition, and the endpoints that will be used to assess that will be endpoints that for the most part can be used in a clinical setting. Next slide. Activities that are considered non-responsive to this funding opportunity include the development of animals and ex vivo models for the purpose of understanding disease etiology or the identification of CNS drug targets. So as Becky mentioned previously, this is probably better for the R01 space. Discovery and development of biomarkers or applications with the primary focus of developing pharmacodynamic biomarkers. I should point out that NINDS does have a biomarker program with a separate set of funding opportunities for those who are interested in those sorts of activities. Other non-responsive activities would include those that fall under one of the other two IGNITE funding opportunities that we're discussing today, which in general include all studies to identify, optimize, evaluate, and develop a potential therapeutic, such as assay development and screening that Becky's had already discussed, as well as efficacy studies and pharmacokinetic studies that Chardell is about to go through. Discovery and development of devices, device-drug combinations, surgical procedures, diagnostics, or rehabilitation studies are also non-responsive, as are human subjects research, with the exception of studies that meet the CFR criteria for exemption 4. Okay, I will turn it over to Chardell.
Thanks, Jules, and good afternoon, everyone. The last FOA in our IGNITE suite of opportunities is PAR 21-122. The Neurotherapeutic Agent Characterization and In Vivo Efficacy Studies FOA provides funding for studies to demonstrate that early-stage neurotherapeutics have sufficient biological activity to warrant further development. So therapeutic agents include small molecules, biologics, or biotech-driven products. We use a combination of parameters, including target engagement, PK, and in vivo efficacy studies to determine the feasibility of the candidate therapeutic agents. Necessary entry criteria that you should be sure to illustrate in your application include novelty, such as new knowledge around targets, mechanisms, pathways, or therapeutic indications. Our projects should aim to develop or repurpose therapeutics that are significant improvements over existing therapeutics to treat neurological disorders. So we're looking for something above a status quo. There must be a strong biological rationale for the intended approach, along with preliminary data or literature-based evidence supporting the feasibility and importance of your proposed approach. Preliminary data should be rigorous and from well-designed experiments. For your reference and convenience, there are links within the FOA that outline some of the most critical elements to meet this criteria of well-designed experiments. The presented evidence within your application should show or support that the therapeutic agent has the potential to be therapeutically viable, and also the robustness of your PD measures and/or in vivo efficacy models. Projects should include a description of the unmet need for the therapeutic agent and a clear justification for how the findings from these studies are relevant to treatments for disorders that fall within the NINDS mission. Projects should also explain how the candidate therapeutic will be optimized in advance towards later stages of discovery once the R33 phase is complete. Next slide, please. So some examples of activities for the R61 phase include those listed here. And while this is not an exhaustive list, the next two slides taken together will provide a rough draft of where you could place your studies within your application. So activities usually found in the first phase are preparation of the therapeutic agent to support your proposed activities, characterization of the agent. This could include, for example, confirmation of purity, stability, PK properties such as in vitro absorption, distribution, metabolism, and excretion, in vitro potency and selectivity, as well as degradation products and process influence. Limited SAR to optimize the candidate therapeutic agent for in vivo efficacy testing is allowed. Studies to develop dosage forms to support PD measures and/or in vivo studies. Pharmacokinetic studies to determine direct or indirect therapeutic agent levels. Studies to confirm that the therapeutic agents reach and engage the target site either directly or indirectly, and at a level that exceeds in vitro pharmacological potency over the desired period is allowed. Studies to confirm selectivity of the therapeutic agents. Limited studies to identify potential pharmacodynamic biomarkers specific for the purposes of developing the therapeutic agent to be tested in the application are allowed. And also studies to inform design and refinement of the PD measure and/or in vivo efficacy models and testing procedures. This may include examination of model variability to estimate sample size, calibration of the model according to positive and negative controls, along with age, time course, and window of disease that are relevant for therapeutic testing. Next slide, please. At the end of the R61 phase, there are some criteria that should be met to transition to the R33 phase. And again, we have these milestones set up. So if you meet your milestones, you should see successful completion of all necessary preparation and characterization of your proposed therapeutic agent, such as any chemical synthesis or scale up that's needed to provide material for your proposed project, all of your in vitro or in vivo potency studies, in vitro organ tissue or cell culture systems, all PK studies, any design, refinement, and validation with PD markers and/or in vivo efficacy animal studies, using, of course, appropriate positive and negative controls, and demonstrating feasibility of conducting therapeutic agent testing. You should also have a further refined and detailed in vivo study design, such as in the animal model, that meets the NINDS rigor guidelines and will allow demonstration of dose and exposure response. Examples of some activities for the R33 phase, but are not limited to, include pharmacodynamic and/or in vivo efficacy studies with chemically and biologically characterized therapeutic agents, dose response activity with the intended route of administration relevant to future clinical studies, studies correlating to pharmacokinetic and pharmacodynamic measures, any preliminary studies to assess early safety. Of course, this is not including those IND-enabling toxicity studies, and then validation and replication studies to confirm your observed results. Next slide, please. So there are some activities that are either not in scope of the IGNITE program or fall within another IGNITE FOA. And some examples of activities that are non-responsive to this particular FOA are shown here. And again, you've heard this earlier. We're asking you pay special attention to these, as applications that include these elements and apply to this FOA will be considered non-responsive. That means your application will get withdrawn and not reviewed. We're asking that you see the FOA for the complete list. These are a few items that are included. And we understand this can be a confusing point for applicants when they are deciding which FOA to submit their research. For this particular FOA, non-responsive studies include anything that has development of de novo animal models, assay development, discovery of novel therapeutic agents. These are covered by companion IGNITE FOAs. Anything that has discovery and development of risk, detection, diagnostic, prognostic, predictive, or prevention biomarkers. And again, we say, although the use of existing biomarkers is appropriate. Applications where the primary focus is on developing pharmacodynamic biomarkers are non-responsive. GLP toxicology studies, any IND-enabling studies with optimized therapeutic agents are non-responsive. Again, you're past IGNITE if you're there. Discovery or development of devices, any device-drug combinations, surgical procedures, diagnostic or rehabilitation strategies are non-responsive. Studies of disease mechanism or disease target identification, again, are non-responsive. And then we have the human subjects research criteria, which Jules will discuss in a few slides. So any human subjects research, except for research that meets the exemption 4 criteria, are also non-responsive. Thank you.
Great, thank you, Chardell. So I'm going to come back and just give you some general tips, and these apply to all 3 FOAs. So please contact us in advance. We know this mechanism is a little different from like an R01. We'd much rather you talk to us in advance than have some confusion that prevents you from scoring well. We're happy to help, and the best thing to do is to send us an AIMS page, and we can then make sure that you're at the right institute and looking at the right FOA before we set up a call, just to save your time. Please make sure you include rigorous study design. If you've been paying attention to NINDS at all in the past 10 years, you'll know that we've been taking rigor very seriously. This is even more important when it comes to translational research, because hopefully you're building a package that will support a lot of future activities, including some expensive activities, maybe even clinical trials down the road. So it's really important that the data package be rigorous and strong. If you have to make a decision between running more compounds and running fewer compounds in a more rigorous way, choose to do things rigorously. You can see our notice here, and by rigor, I'm not just talking about what you propose to do in your application, but also the preliminary data, the supporting data. Very important. The next thing is have a multidisciplinary team, and note the multidisciplinary review. So the review panels for these, we have expertise not just in the disease, but also in assay development and medchem and animal models and every step down the road. We have clinicians, we have industry, we have academic scientists all on the panel. And we do this so that we get a really good multidisciplinary look at your application, and you will get 4 or 5 reviewers. And this is on purpose so that we get all of these different perspectives looking at your application. So know that that's your audience. And the best way to write a strong application that satisfies a multidisciplinary review panel is to have a multidisciplinary team yourself. who can help you write an application that's going to, answer questions for all of these different panelists, okay? Know your audience there. Strive to increase the diversity of your team. So we've added stronger language to these FOAs when we reissued them. I think you'll notice a lot of NIH FOAs are now having stronger language here. Hopefully you'll notice that this is a tide that's hopefully turning. Perhaps, we won't talk about if it's too late or too slow, but it is something that's coming. So please help us fund more inclusive science and think about the diversity of your team. Discuss intellectual property, especially for therapeutics. This is something that people don't always think to do if they're coming from an R01 space, but it is important. You do not need to have patents filed at this point, but you do need to be convincing reviewers that you're thinking about it. Convince reviewers that if this is successful, it can move forward and it won't just fall off a cliff because you haven't thought things through. If you can get a letter of support from your tech transfer office saying that they'll work with you, that's about what you need at this point. Again, don't feel obligated to have patents filed for Ignite. Just be thinking about it. Have a therapy development plan. Again, convince reviewers that this has a path forward. If these 3 years are successful and at the end you meet your goals, then what? Again, we don't want to fund something that's going to fall off a cliff. We want to fund something that has a path. So talk about what that path is. Again, you don't need to have CROs lined up. You don't have to have, really everything planned out, but you do need to convince reviewers that you're thinking about it and that this has potential and that you have people on the team who will help you think about it and prevent problems before they happen, or, basically that you're thinking ahead. All right. And this last bullet, this is also new for the new FOA. So I'm going to point this out. It's buried deep in the FOA. You might not have noticed it. So I feel it's fair to warn you. We will be requiring NIH approval for no-cost extensions, not because we don't want to see people use them, but just because we want to understand. It's really for analysis and tracking purposes. Because we see a lot of no-cost extensions in these mechanisms, and we just want to understand why. It's not our intent to stop projects. It's just this is how what we have to do in order to get the data to do an analysis. So be aware of that. And all right, and then some more general tips. If you are SBIR eligible, if you're a small business, you are eligible to apply to Ignite, but I would recommend you consider the SBIR program. The reason is NIH sets aside funds for this program, and if you are eligible, then you're only competing with other small businesses for this pot of money instead of competing with the general pool for the general Ignite program. Have go-no-go milestones. I think I've already told you about this, but notice that this is a responsiveness issue. Applications without milestones will be withdrawn. That's the structure of the mechanism, right? I told you at the beginning this was really important for this mechanism to work. So if there's no milestones, there's really no way for us to use this phased mechanism. You need to clearly demarcate the R61 versus the R33 activities in the timeline. Again, this is a responsiveness issue. Pay attention to non-responsive activities. We've already told you that. And pay attention to human subjects research designations. And with that, I'm going to turn it over to Jules, who's going to tell us a little bit more about that.
Great, thanks, Becky. So we wanted to briefly touch on the terms for human subjects research, since this can be a point of confusion for some of our applicants, especially when it comes to using human samples or patient-derived specimens. So human subjects research is defined as research involving a living individual about whom data or biospecimens are obtained, used. Oh, sorry. I don't know if you guys can hear me. I will start over. So research involving a living individual about whom data or biospecimens are obtained, used, studied, or analyzed through an interaction or intervention, or where identifiable private information is used, studied, analyzed, or generated. Now, within the designation of human subjects research, The Code for Federal Regulations include 8 different types of exemptions, and here we're specifically talking about Exemption 4. So what is Exemption 4? It involves the collection or study of data or specimens if the sources are publicly available or the information is recorded such that subjects cannot be identified. But the key is that Exemption 4 is for secondary research only. If the biospecimens are collected specifically for the research that you're proposing in the grant through an interaction with living individuals, then it's considered primary research. So overall, depending on the specifics of how your samples are collected and how they are coded or de-identified, will determine whether it qualifies as human subjects research at all, and if so, whether it would meet the Exemption 4 criteria. So the NIH's Office of Extramural Research provides a number of resources through their website, and we've included the link here, and we would highly encourage you to visit. Importantly, if you're unsure about the designation of your research, we highly recommend reaching out to your institute's IRB or your institute's human subjects officer. You can also email OER at the address provided here. Can you go to the next slide, please? This is just the homepage for the website that I was referring to. In particular, I wanted to point out that they have a decision tool which guides you through a series of questions, and we found this to be quite helpful for our applicants. So we would definitely encourage you to look at all of the informational resources on this page. And so with that, I think we'll go to the next slide and start going through the questions.
Great, thank you, Jules. Yeah, so feel free to keep the questions coming. I see some are in the Q&A already. If it's something that's specific to you and your project, though, please just contact us, send us your AIMS page. It's just hard for us to answer really specific questions without seeing the AIMS first. Plus then we can use this time for things that are applicable to the broader group. Jules, do you want to get us started?
Sure. So a couple questions related to the preliminary data. So essentially, how much preliminary data is expected at the time of your original submission and what's needed here?
Yeah, that's a good question. And there's not a hard answer to that, or a clear answer to that. It really, the goal is to convince reviewers that what you're doing is feasible and that it's worth doing. And so you need enough data to convince them of that. And that could, by the way, be, literature data. It doesn't necessarily all have to be, done in your lab. So just give reviewers confidence of those two things, I think, is really what you need to be thinking about. Next one.
What is better for the R61 phase? Assay development or a developed assay needing optimization for high throughput screening?
We support both. Both are probably important. It really depends on the state of the assays that you have or what you feel you need. I don't think there's, I think both are important.
Could you clarify R61 to R33 transition? Will we have to resubmit another application after 2 years to request a separate funding for the R33 phase?
So the way it works, you will submit paperwork, you will submit a form. It is not like going to peer review all over again, but you will submit a form that says you feel you've met your milestones and you want to progress, and then we will have a discussion. And we'll set up a call with you and talk through your data. And then, if we agree that the milestones are met, then we process your R33 paperwork.
I should point out again, just as a reminder, that your original application does need to propose both phases, though.
Yes, absolutely. Good point. Thank you.
Okay, next question. How does the Blueprint program for biologics differ from the Create Bio funding program?
Jules, would you like to take this one?
Sure. So both programs support biologics. Create Bio is the existing NINDS program to go down that path, but that program is sunsetting. So the current funding opportunities are not at the moment planned to be reissued. We have a Blueprint Neurotherapeutics program that is currently for small molecules. And as Becky mentioned previously, this has recently expanded to also include biologics. So you can do the same activities that you could do and create in BPN. The structure is a little different. You have a lead development team that is a lot more NIH staff involvement and consultants that will sort of guide the milestones. And you could also make use of NIH contract resources, CROs, or you could still use your own. But this is a very new program. We're probably going to have a webinar sometime in the next month or two, and the first receipt date is in August. So stay tuned for more information, and please feel free to contact us if you have additional questions about that program. But again, that's a later stage program. So you would do stuff in Ignite in order to get to the entry criteria for that program. Okay, for PAR 21-122, so our efficacy PAR, if we have already identified a lead compound with demonstrated efficacy, would it be responsive to propose to test efficacy of related molecules?
Yes, you could do that. Although if you feel like you have a strong proof of concept with your starting compound, you could look at Blueprint. I think we'd have to have a discussion about whether or not Ignite or Blueprint was the best fit for your particular project based on, the data. But that would be within scope, I guess, is the short answer.
Mm-hmm. Okay, also for PAR 21-122, some of the criteria seem designed for small molecules. What about biologics? So AAV doing PK may be a bit difficult, for example.
Yeah, we absolutely support biologics, and I apologize if I made it sound like this was a small molecule only program. All of both of the 122 and the 124 support both small molecules and biologics. So maybe instead of calling it PK, you're looking at distribution versus time or something. But absolutely, we support biologics.
If a lead compound has already been licensed to a startup, can it still be included in these applications, assuming the company is on board? If not, can derivatives be included, or is the whole family ineligible? And sort of a follow-up to that, can a startup that has licensed a novel compound apply to these programs to further develop the compound?
Right. So yes, a startup, like I said, if you're a small business, you are eligible for Ignite, but I would encourage you to look at this BIR program instead. But in terms of licenses and patents and stuff, you do need to have freedom to move forward, and you need to convince reviewers that there's a path forward. And that could be that you're developing something that somebody else owns and you have their buy-in and they're okay with that, then that's okay. We just need to know that there's a path forward.
I should also emphasize that in addition to the SBIR program, we also have an STTR program,
Yes.
which is more of a collaborative effort between academic and small business. So the small business is still the primary applicant and where the money goes to, but the PI can be from the academic institute that's collaborating with it. So if this is something that you think would qualify for, then definitely reach out to our small business individuals.
Yes, thank you.
Okay. Also for PAR 21-122, we have a lead nanoscale drug carrier that, when loaded with 3 different small molecule drugs, ameliorates our animal models. Can we use this to compare which of the 3 cargo drugs in our nanocarriers is most effective?
This sounds like a very project-specific question. Why don't you send us an Ames page and we'll talk about it? Because it really gets to the goals. Is the goal that you're developing a therapeutic, or are you understanding, mechanistic stuff if you're comparing? I guess I would need more information. So
Yeah.
Send us an Ames page and we'll talk about it.
So I guess a more general question from that is, can you use our in vivo efficacy FOA to compare different therapeutics?
Yeah, that's a good point, Jules. Yeah, yes, you can. And certainly we see people do that. You've got 3 or 5 candidates and you're going to use R20 or 61 to pick the best one. And that's totally appropriate. We see that all the time.
Okay. While target and mechanism of action are not allowed, a previous slide said selectivity is allowed. Would you clarify, please?
Yeah, so if you're developing a therapeutic, you're probably going to need to know that it hits your target and not other targets. And that's part of therapeutic development. And that's okay. If you're looking at a bunch of different things because you're fishing and you're understanding the disease and you're not sure which of these targets is important for the disease, that's more mechanistic. And if this distinction is sounding a little fuzzy to you, then we can certainly have a discussion about your particular project to make that more clear.
Okay. Could you please provide the link from Office of the Director on diversity of scientific applicant teams? So I should note, yeah, so it's in here. And then within the FOAs themselves, there's also a link to notices that have additional links within them to all of the relevant information.
Yeah, I don't know if you can see my cursor, but it's like the middle bullet here, OD 20031.
Okay. Can you discuss the budget of each phase of the grant?
Yeah, so again, it's $750 total direct costs for the full 3 years, and then no more than $499 in any given year. So however you want to divide up the time and the budget is up to you as long as you stay within those 2 rules. You cannot have more than 2 years in any one phase, and no more than 3 years total. So it's up to you how you make that distribution work.
Is therapeutic nanotechnology responsive to PAR 21-122?
Potentially. Again, send us your aims and we can make sure look at it for sure. But that sounds like a biologic technology, most likely.
Mm-hmm. Sorry, going through these as they're coming in.
Yep.
We have significant preliminary data of our compound in ophthalmic indications, but want to determine if the small molecule works in other diseases. Is this a fundable effort?
So if it's within NINDS mission, potentially, but it would need to be an NINDS fit or NCH special zone there. Again, if you send us an aims page, we can help you make that determination.
Is there an equivalent program for initial human subjects testing of novel therapeutics? Does not meet exemption 4.
Okay, so if you're testing your drug or your therapeutic small molecule biologic in a human subject, if you're looking to do like intervention trials, Blueprint does support that for very early work, but I don't think they're going to accept it if that's all you have to do. But if you come in earlier and they're getting to that stage, they will support it. As beyond that, there are other programs in our Division of Clinical Research. If you want to take a look at those, just if you Google NINDS DCR, you'll find their webpage, and they have other programs for later stage clinical trials.
Okay, really, is combining novel animal model while developing a novel biologic acceptable as a single application?
That would be a challenge. So no, it's not. And we divide them up because it would be really challenging to do a good, thorough, rigorous job of both in 3 years. So we've separated them out intentionally. You can develop your animal model, do all of the validations that need to be done in one grant, and then separately do your biologics development and testing in another grant.
Okay. Do plans to work with CROs disqualify from having a multidisciplinary team?
I don't know why a CRO would disqualify you. No, you can certainly work with CROs. Plenty of people do.
Yeah, so for the multidisciplinary team, we want to make sure that your project has all the necessary expertise to perform the activities well. So if a CRO is capable of doing that, then that would still satisfy that criteria.
Yep.
Could you say a little more about a university-based person working on a small molecule owned by a startup?
Yeah, so you can do that if you have the startup's buy-in, and I would definitely make sure you had a letter of support from the startup. If that startup owns it and you're testing it, make it really clear in that letter that they're supportive of you using it and they're going to provide you, if you need it, that compound. And also, what's the path forward? If Ignite is successful, does the startup then take it forward from here? You want to give reviewers confidence that there's a path forward. We don't want a situation where we fund a grant and then the company goes away and decides they're not interested, and halfway through the grant, there's no way to move forward. So anything you can do to make it clear that that's not the case. But it's certainly allowable as long as there's a path and that's clear. Is there anything else?
Yeah, sorry, I'm reading one question right now, but it's very specific, so I'm going to have them contact it.
Okay.
But I think there were a couple questions that came in prior. Let's see. One question is asking for whether assay development and neurotherapeutic agent identification from a natural product library in neuroblastoma is appropriate for 21-124. And the answer is yes. So neuroblastoma is within NINDS submission. So that would be okay.
Yeah, most of the time. Just do send us an AIMS page though before you submit a full application so we can just verify, because NCI does take some of that, and I wouldn't want you to put forward an application that didn't have a home.
Yeah, and I'll just add that would not be appropriate for NCCIH though. So that's an example where our interests are a subset of NINDS's interests, because a natural product screening against neuroblastoma would be responsive to our interests as they relate to this PAR.
Yeah, thank you. Yeah, and since it would be an NINDS zone, it doesn't matter if it's natural product or not. But yeah, thanks, Craig.
Okay, and the last sort of comment is it would be extremely helpful to have criteria for probe development and drug targets that are accepted by the NIH review panel. So I guess the question is, how does NIH define an appropriate accepted target for a disease indication?
Yeah. So I'll talk about the probe first, though. That would be generally not an scope for Ignite, since that's generally basic science space. But if you're looking for how do you define what target is ready to go for Ignite, it kind of gets back to the preliminary data question. It's a case by case. I can't tell you need to show X, Y, and Z, and you're good to go. It's really, it's a reviewer evaluation situation. So convince reviewers that it's ready for therapy development and that your target is validated enough that it's worth pursuing for therapy development, and that it's a viable target, but at the same time novel and interesting. So I don't feel like I could give you a stronger answer than that. It's just it's not well defined because we want reviewers to be able to evaluate on a case by case basis. Is that our last question, Jules?
It is.
Excellent. All right. Well, thanks for hanging with us. We ended right on time, so that's great. Thank you, everybody, for your participation and for the great questions and for your attention today. Again, we'll be sending the slides out with the links that you can click. This has been recorded and will be shared. So if you missed something or if you want to send it to a colleague, you'll be able to do that. And as always, send us your questions, send us your AIMS page. You can send stuff to Ashley, her things here. My email address and Chardell's and Jules and Craig's are all at the front of the slides. So you know how to reach us if you have any questions. With that, I'll close us out and thank you again.